All right, Ryan Stanfield, Foldax. It's great to have you on the podcast.
Great to be here. Thank you very much, Steve.
Yeah, I'm really excited, Ryan. There's been so much buzz about Foldax and the world's first polymer heart valve. So I'm really looking forward to this conversation. I know you're going to share some results from a study you did, preclinical evaluation and early feasibility of a novel polymer for cardiovascular use.
Yeah, we got a lot of different cool results to talk about. We've been working on this valve for a number of years, working on the polymer. I'll give a background on the polymer. We basically went into it designing a new valve and a polymer valve, and many of us have experience with polymer valves in the past and trying to work on them. But the whole background is we said, hey, we need to have a new polymer, and so we designed a valve. But then designed a new polymer. And so we partnered with CSIRO in Australia to design the polymer.
Okay. So that polymer that was designed, it's a new formula or compound. It's not like a predicate that's being used in like a neurostimulator or some other implantable application.
Exactly. Yeah, it's I would say it's a close cousin, excuse me, to a polymer called Elastion.
Yep.
Abbott uses it on their pacemaker leads, and so it has a lot of use there. And people don't really realize how much cyclic loading is on those pacemaker leads, right? But they do flex and move as they're in the heart, and so there is some translation there of the mechanical properties and durability that's required on pacemaker leads and into heart valves. And so, we tried to leverage that, what we knew about, some of those other polymers and other polymers we've worked with in the past and develop this new polymer. And I'm not a polymer chemist by any means, but, we have some absolutely amazing polymer chemists that are part of our team that have been part of the development of this polymer, of LifePolymer.
Yeah, LifePolymer. It's a siloxane-based polyurethane, I believe.
Okay. Yeah, it's a siloxane polyurethane urea.
Okay.
Specifically.
Okay. Yeah, I look forward to digging into that. Before we do that, why don't we talk about some of the unmet needs in the mitral valve, heart valve replacement space that you're tackling?
Yeah, there are. That's I'd say a pretty deep and potentially a deep topic there. But one of them that we were just discussing that I'll just elaborate on a little bit further is rheumatic heart disease. And rheumatic heart disease is prevalent in developing nations. We've begun a trial and completed a trial of our mitral valve in India, where rheumatic heart disease is rampant. And so what happens, and again, I'm an engineer, I'm not a clinician, but what I can tell you, what I do know is that, rheumatic heart disease destroys the valve leaflet over time. And it's specific to the mitral valve in many countries and many demographics. There are other countries in sub-Saharan Africa, for example, where the rheumatic heart disease will damage more than the mitral valve. It will hit the tricuspid valve as well. And so, but specific to the mitral valve and India, there's a large patient population there. There also is in South Africa. We could go on and explore other areas, but in India, there's a large population with rheumatic heart disease, and it also impacts women as well as men. And so, with the patient population in women, they have rheumatic heart disease, and they can't really repair the valve; they have to replace it. And so they can either choose a tissue valve or a mechanical valve. And the mechanical valve comes with lifetime anticoagulation that they need to be on to maintain valve patency. And there are things that have been worked on there scientifically and clinically to reduce the amount of anticoagulation that patients have to take, but they still have to be on anticoagulation for a lifetime. On the other hand, you've got tissue valves, bioprosthesis, that don't necessarily require anticoagulation. However, they don't last as long. The mechanical valves last a lifetime. The tissue valves wear out and degrade over time. And that's particularly true with tissue valves in the mitral position and with rheumatic heart disease. That just accelerates that degradation. And so, patients, I think in the mitral valve space, they have about 5 years with the tissue valve on average before it needs to be replaced. That could go up to 10 years, but still, if you're talking younger patients, especially females that are of childbearing age,
right.
They have to, if they choose a tissue valve, they know that they'll need another valve replacement in the future, and we'll have to go through that procedure.
Okay. So kind of comes down to this durability issue as well as being on the anticoagulant for a lifetime.
Yeah. And there are a lot of things that go along with being on anticoagulation, right? If you're on anticoagulation, you can't, or you shouldn't be conceiving, getting pregnant. And so that's the appeal to this childbearing age population is having a valve that has both sides, as both the durability of a mechanical valve, but also the reduced requirement for anticoagulation. And that's, what we're going for with the polymer.
Okay. Yeah, let's get into the LifePolymer a little bit and talk about some of the, the properties, the biostability, mechanical properties and such. And I think you might have some slides you can share on that. If you'd like to pull those up, that'd be great.
Yeah, happy to. Let me see if I can get this the best way to do this. How about we try this? I'm going to start. Let's see. How's that?
That's good. That's really good.
See that okay?
Yeah.
Okay, so I'll start with, this is just kind of a reiteration of what I just spoke about, right? It's not saying that tissue valves or mechanical valves are better than which one is better. I mean, there are preferences. They both have, they're both costly to make. The durability on the mechanical valves is greater, but the quality of life on the tissue valves is greater. And because of that anticoagulation. Also, when valves wear out, and if there's an issue with the tissue valves, because they have a fully opening orifice, you can do valve in valve. That's a procedure, when they can go in and actually put a transcatheter valve inside of a surgically placed valve when the valve wears out.
Gotcha.
If that makes sense.
Does.
So that's an option with some of these tissue valves, right? You can't really do that with this mechanical valve.
Okay.
Because the hinges are right there in the middle. Can't put a valve inside of it. Happy to talk about the polymer. And I'll go through some of this pretty quickly. This is a really good graphic that I'll put on the screen. It talks about the development of polymer valves over the last 70 plus years, 75 years now, starting off with PDMS and PTFE and then expanded PTFE. Going into a bunch of new polymers that you see on the left side of this clock here. And what we're focused on, first and foremost, is solving this list of problems or demands that come through valves, right? They have to have certain physical properties, be biostable, creep resistant, so they can't, have plastic deformation over time, be hemocompatible, not calcify, And that's a big challenge with children, actually.
Mm-hmm.
Their calcification profile is different than adults.
Hmm.
And they have to be biocompatible, of course, meet ISO 10993. And then, of course, you want something that you can manufacture, right? You have a supply chain for that you can, build with confidence and be, cost effective as well.
That's a great chart. I love that chart on the right.
Yeah, it's great. It comes from this publication that came out 2 years ago by Rezvova in the International Journal of Molecular Science. So it's a great little diagram. So it's specifically with polyurethanes, right? They have hard and soft segments. They're used in a lot of biomedical applications, but a lot of other applications that are not just biomedical, right? They're used all over the world,
right?
In a number of things. There aren't any chemical crosslinks, so it's a thermoplastic. And so, you have these little kind of hard and soft domains, of what's inside of the polyurethane. And then, the polyurethane has tunable mechanical properties because of this, both hard and soft segments, right? You can focus on mechanical strength with the hard segments, and then kind of the flexibility with the soft segments.
Right.
And so there's low modulus polyurethanes that have been used, but they can degrade in long-term implants, and those are kind of seen here with some of these images from a 1991 paper. And so there's a lot of different focus that's been put on, development of polyurethanes and treatment of those to make them last longer or not. But you can see that there is pitting and cracking that can happen with them as you look at it under scanning electron microscopy. And so, in the mid-90s, they started to improve polyurethane, and that was developed by this group, CSIRO, in Australia, incorporating siloxane as the soft element. And they also incorporated a compatibilizer. So that permits the siloxane and the urethane hard segments to react. So, it creates this high molecular weight polyurethane that resists degradation. And so part of that's using a PHMO in the chemistry and the manufacture of it. And so, Elastion or Optum are these copolymer silicone polyurethanes that were developed. And I mentioned earlier, they're used as pacing leads.
Yep.
And Abbott has a great report on their website. It's their product performance report that talks about that. I won't get into any details, but Abbott's got a lot of material on that.
Yep. I think they bought that, didn't they?
They did, yeah. Yeah.
Okay.
Yeah. Bought or licensed for that specific application. So the LifePolymer itself that we've worked on and worked on hand in hand with the group at CSIRO in Australia, we use that same kind of hard and soft segment structure to make something that's specifically degradation resistant for these leaflets. So it also incorporates urea groups, linked macrodiol, and then another chain extender, and to get these properties that preserve stability, but is also kind of creates the best of both worlds. Improving the flexibility for hemodynamics, and then resists creep, resists tear propagation, etc.
Interesting.
And so this is just tested the polymer that we've done, LifePolymer, looking at calcification. And so you can take a strip of this polymer and put it subcutaneous in a rabbit. That's a standard test that's done according to the 10993 ISO standard, and look at what happens to that over time, after 6 months, if there's any calcium on it. And we also have had, this is on the right, you see actually one of the patients that we've had explanted. It doesn't occur very often, but these explants are, worth more than gold to us. And we can look at the polymer, and, you can see here as well that there's no trace of calcium on that polymer leaflet after it's been in for quite a while.
Interesting.
Other tests that we've done and this again specific to the polymer, you can see, so there's what's called an ex vivo shunt study that's done. The world experts on this are at Oregon Health Science University. And this is published about 4 years ago by Chris Jenny and others in Advanced Nano-Biomed Research. References down there at the bottom. But we did this test. So there you've got this ex vivo shunt that comes out of the animal, and we compare it to EPTFE. And EPTFE, everybody knows, is a very non-thrombogenic material. It's a really great material, but this is kind of showing them head to head. And LifePolymer is actually much better than EPTFE as a reference.
Wow. By a lot, right?
Yeah. Yeah.
It says a lot right there.
Yeah, right there. So, there's a lot. So this is just kind of the bench testing, I would say, in vivo and ex vivo and in vitro tests that we've done on the polymer. There are a lot of other polymer properties that are published in this specific study that Chris Jenny published, and others that talk about the mechanical properties of the polymer as well.
Okay. That's a compelling chart right there.
So I'm happy to keep going and talk to you about the kind of the CAD modeling, the finite element analysis that we did when we were designing.
Yeah, I appreciate that. Yeah, let's go into some of that. Definitely, and certainly anyone that looks on your website too can see that you do a lot in that area. So, and it's all in the public domain. So yeah, if you can share a little bit about the modeling and the robotics that you use in the manufacturing process would be wonderful.
Yeah. So there's quite a bit to say there. Just the design of the valve. The valve has kind of two main structural components. There's a frame, right, that provides the structure for it, and then there's the leaflets. And so I'll talk about the frame first. So this is specifically an aortic design, but the principles are equally true for a mitral valve design. We do have specific designs for aortic versus mitral. We don't just flip it over, right? We don't just use the aortic valve in the mitral position. The valves, the frames, and the leaflets are optimized specifically for their intended application, intended use environment. So, the part of this, we can take this and optimize this around, how well the commissure posts. That's what we call these tips here, where you see the red peak.
Yeah.
There will be some deflection there, and that will help the valve leaflets coapt, and when the valve is closing, and it also provides kind of a spring force to open them back up, right, when that time comes. And so you see that here. This is just a finite element analysis. After things have been optimized and designed, we can look at what the stress distribution is here, and where polymer has an advantage over tissue is, we've got a flexible surface, and we have, particularly with LifePolymer, we have what we can assume to be a homogeneous a polymer material, right? A tissue is not homogeneous, right? It's almost a composite material, right? It's got fibers in it and directionality to how the stresses and strains. And, this is what we can assume to be an isotropic material, especially at this level. And so that makes the analysis and the design, I wouldn't say easy, but it makes it a little bit more straightforward.
Yeah. I noticed that on the flexion areas where you probably hit, I know at least on a TAVR, kind of going back to TAVR, the flexion zone is where there's the most stress, and that happens to be where it's sutured to the frame, and so that's a weak point. But I don't see any red near the flexion zone in this case. Am I reading that right?
Yeah, you are. I mean, there are some high stresses around there, right? If you want to pay attention to the pixel that's here right at the commissure, there will be some, I'd say, instantaneous high stresses around that area. But it's, there's also how you design the leaflet and how that will carry the load, as well as how you design the frame. And thank you for pointing it out. I will talk about it more, but there are no sutures in this valve, right? The valve leaflet. And so I'll show that on this next slide.
Please.
We've talked about this. I mentioned, we've got a number of sizes here, and the valve is optimized around or for each size, right? In the aortic orientation and the mitral orientation, you have two very different environments, right? You have the left ventricle pressures on both of them, but the aortic pressure versus the left atrial pressure are very, very different. And so you have a very different pressure gradient, especially as the valve is closing, that you really want to optimize for. The other thing too is, these leaflets are about a third the thickness of biologic tissue. They're about 100 microns thick. And so that reduces the inertia, right? So if you think about it, something opening or moving just in general, right? If you use the same amount of energy, these will open, much faster than a tissue leaflet would.
Gotcha.
Just because of that inertia being reduced. And so that can lead to a couple things that I'll talk about later, but it can lead to basically a more efficient valve.
Makes sense. Yeah. Less work. Right.
Yep, indeed.
Yeah.
So we run these valves, right? We test them on the bench. I talked about testing them in animals. We test them on the bench. There's, the ISO 5840 standard has a number of tests for heart valves. One of them is durability or accelerated wear testing, AWT. And, there are companies that make accelerated wear testers specific for valves because it's such an incredible test that you want a really high reliability machine that can also test the reliability and durability of your valve. And so you have to test it up to 600 million cycles according to the new standard, with a sample size of at least 5. And so, essentially what we show is, over 600 million cycles, this is the equivalent of, somewhere between, I'd say 15 and 20 years of use, depending on what you say is, a normal heart rate. So it takes almost 2 years to run this test in an accelerated fashion. But what we see here is that, our pressure gradients stay pretty stable throughout that, and our EOA, that's the effective orifice area, right? The valve is opening up to, that stays constant throughout this time. And, this is an aortic valve, same thing. And so you see them here kind of side by side. The one on the left is the Tria valve with LifePolymer, side by side, looking at the Edwards Paramount valve. That's a surgical valve. It's not a transcatheter valve. But you can see, the effective orifice area and how quickly the Tria valve opens and closes. It does. It's hard to say it's much faster. They're on the same cycle, right? Same kind of beats per minute, but you can observe that that difference.
Definitely.
looking at, us implanting it. This is an ex vivo experiment. We put this in an animal heart on the table. And so you can see in the top left is an aortic valve. And then the bigger one that's on kind of the bottom right, that's a mitral valve. And so you see both of them opening and closing. And clinicians, they'll look at this and they'll be, I mean, everybody, you look at this compared to other valves and you can see right through the valve leaflets, right? So it's a little bit different.
Yeah, it's transparent. Yeah.
Yep.
That's very cool.
So Steve, I'm happy to talk a little bit more about and get into the manufacturing of this and what we've done there. The great thing about polymer, like I said before, is if you know how to use it and work with it, it lends itself very well to automation. Right, the tissue and the mechanical valves, right? Those are individually made valves that have to be placed onto a frame in some sort of mechanical fashion with some sort of fixturing, right? And not just a fixture to place the leaflet, but some way to fix the leaflet in place, either a hinge on a mechanical valve or or sutures or something like that to tie down a leaflet that's made of tissue onto a frame.
I know anytime that you get involved with sutures and a frame, a stent frame, vascular frame, that's a real challenge from a scalability standpoint. Right?
Yeah.
It's hard to scale that, and it's hard to really, that's a hard job to keep someone on for more than a year, probably, right? I've heard of some of these stories of both of those situations.
Yeah, and it's a specialized skill too, right? I mean, just the dexterity that's required for it. We joke about that. We would early on, and some of the people that have been at Foldax have had experience with tissue, and we talk about how tough just tying those knots, right? Burning that suture, putting the suture into a needle, and it's some highly precise stuff.
Right.
And and not even to get into any of that, but when you have a human doing the work, you're subject to human error. Right. And so not only is it tough to scale, but it's also you've got repeatability challenges and error reduction challenges as well.
It's scrap rates. I mean, you spend I don't know how long per valve suturing, but I imagine it takes a long time, and then at the end, you have a defect and you have high scrap rates. So yeah, there's lots of challenges with suturing polymer components, covers, etc. To stent frames. Yeah. So let's talk a little bit about an automated or robotic method that would be fascinating.
Yeah. So I'll get into that. Let me kind of preface that with a couple of other things.
Yeah.
Right. And we've talked about this LifePolymer material, that we have. What we do is we actually do a dip cast, and, I'll show that a little bit further here. But we dip cast onto a mandrel, and that forms the leaflet.
Yeah.
And that mandrel is generated by these leaflet designs that have been optimized with FEA and other computational techniques.
Interesting.
So that eliminates humans in the production process and go with robotic or automated manufacturing, which is totally scalable, right? You can put that anywhere. This one's specific to our mitral valve. This slide kind of shows the LifePolymer leaflets. This sewing ring that's put on, and these 3 LifePolymer leaflets, they're not individually what I want to say, cast as a sheet and then cut out or anything like that and then sewn on or bonded on. All 3 of these leaflets are essentially a single piece. What I say here on the slide as a unibody is we take the frame and it's dipped into the polymer and retracted, and that creates all 3 leaflets simultaneously.
Okay.
And so it's essentially a single piece. And I have a video to talk about that, but
Before you
part
go,
of
can you go back to that real quick?
yeah.
We talked about the world's first polymer heart valve. We're not just talking about the leaflets. I see the frame is peak,
Yep.
right? And I
Yep.
see the porosity of there's some kind of expanded PTFE ring around it. Yeah.
Yep.
So this is
It's
a
full
true,
polymer,
right, full
right?
polymer. I didn't realize that before our call. So when you showed that slide, I'm like, oh, wow, we really are a polymer heart valve. I thought it was,
All parts polymer. Yep. You're
yeah,
right.
I thought
Yep.
there'd be some nitinol in there, but go
Thank
ahead.
thank
Yeah.
you for bringing that up.
Yeah.
But our surgical valves are full polymer, polyether ether ketone frame,
Yeah.
right? PTFE sewing ring, and then of course the LifePolymer, the siloxane polyurethane urea leaflets.
Yeah. Very interesting.
And if you look at that and you kind of compare some of this stuff between mechanical valves and bioprosthetic valves, right? The life polymer, it resists the calcification that is present and can happen in bioprosthetic valves. And like you mentioned, this is full polymer, right? So there's actually no animal tissue used, right? So it eliminates variability and risks that are associated with animal products.
Mhm.
Any other ethical concerns, supply chain concerns, right?
Right.
I mean, you have to grow animals to get to get those tissues. And then it's designed to maintain function and have the structural integrity. That's hopefully positive over kind of some of the things that have been observed with bioprosthetic valves.
Right.
And comparing it to mechanical valves, there's no hinges, no noise. I don't know if you've ever met anybody that has a mechanical valve, but you can hear it clicking,
Oh.
and especially at night. If I've got a couple of friends and acquaintances who will have somebody in their family that has a mechanical valve, and they'll be like, "Yeah, I can hear it across the house at night." It's like a clock.
Yeah.
If you're in the same room with an analog clock that's ticking,
Quick.
right?
Yeah.
You
Wow.
hear the same thing. So there's, we have a tri-leaflet design similar to what a lot of the tissue valves have, and has some advantages over these mechanical valves. And like I said before, you can actually do valve-in-valve procedure with something like this that you couldn't do with a mechanical valve, traditional
That's
mechanical
a
valve.
huge advantage right there too. Yeah.
So now, Steve, to get to the moment
Yeah.
that you've been asking about leading up to this, and that's our automation and robotic manufacturing, and how we've taken this polymer and polymers, I should say, right, including the PEEK and the PTFE, to make low cost and scalable manufacturing solution. So this video is available on our website if anybody wants to go and see it
Yeah.
there. But I can kind of, narrate a little bit. You saw it going there. We have in-process inspection that looks at everything. That showed the dipping of the valve. And then we do this trimming step where we trim the free edge, right? I talked about that being a full unibody leaflet. And there you go. You've got a valve, and then it goes in for some inspection. We look for material consistency, we look for defects, etc. And we can do that all in an automated process. And we have machine learning algorithms that we're implementing and continuing to update to improve our process over and over. But with this material, we can have really highly held tolerances, and that means high repeatability. We do polymer batch testing as well, looking at every batch of polymer that we produce for consistency and repeatability, and making sure that we have all of the what do I want to say? All of the specifications met that need to happen for a specific valve.
I did a podcast with somebody about a year ago about dip the dip coating process over a mandrel,
Mm-hmm.
TPU and other materials. And I asked him, is it challenging to scale that? I said, yeah. He's like, think about the latex gloves. That's a huge building, right? And it's very challenging to scale a dip coating process. But unless you have a robotic solution like you have, I suppose, right? And that's the difference. I mean, even if you had a 10 up situation where you're dipping, it's still not really scalable. So you've taken a lot of the labor out of it and made it a little bit a more repeatable process.
Yeah, definitely. It's hard for me to quantify exactly, but I will a little bit with this slide, right? If you just
Yeah,
kind of compare things back and forth, looking at bioprosthetic, you're
yeah,
looking at tissue valves. Those are a lot of steps. I mean, just the suturing alone that you mentioned, right?
right.
So this is we've tried to kind of baseline and say, okay, one valve, like one surgical valve versus one polymer valve. The polymer method that we use, it's significantly lower in the number of steps, the number of people it takes,
Mhm.
the time it takes overall. Here we have specifically the number of stitches. So we do have stitches in ours. We actually have a robot that sews that sewing ring onto the frame.
Gotcha. Yep.
Um, and but it can be done manually. But I'm just saying, like, there's a couple of stitches that are done there compared to how many you need for a tissue valve. And so, it reduces the number of days, production days, production weeks that are required. And it actually takes a lot less time to train somebody, right? I don't need to go and find specialized people
Right.
as much. My manufacturing floor consists of, a handful of robots and a handful of people
Yeah.
that can run the robots and, it's not full automation, right? I don't dump peak in one side and LifePolymer in one side, and then a finished valve comes out the other side. We're not to that point yet,
Yeah.
but eventually we would like to be.
That's such a compelling chart right there. 130 steps to 30.
Yep.
And everything else goes along with it.
And eventually, with this kind of scale and what's happening, I don't know. You brought up a good point with, the coated gloves that you can buy at the hardware store,
Yeah.
right? And bring it, I don't know if we'll ever be making that quantity of heart valves that they make with those
Yeah.
gloves. Hopefully people won't be, just burning through these and then throwing them away
Right.
like they do with the gloves.
Yep.
But what it does, it actually lends itself to, personalized medicine in the future, where we could eventually take, a CT scan of a patient and their heart and customize the mandrel and the frame specific for them through rapid prototyping, through other things that we have in this validated manufacturing methods that we have with the polymer to make a custom valve specific for, a pediatric or an adult patient.
Mm-hmm.
And that's also a big benefit as well.
Interesting. Yeah. Personalized.
That's what we see as the future of. I mean, everybody talks about that as the future of medicine, right? Personalized
Yeah.
medicine, and we think that it would be possible with this kind of scalable manufacturing.
Right. I can see the model that you've set up to design and manufacture lends itself well to a personalized when you're using the 3D software, FEM software, and things like that as well to model these things. I can see where you can be more personalized.
Indeed.
Yeah, interesting. Yeah, I was going to ask you about this, whether this is a 1-year clinical trial, right?
Yes. Yeah.
Perfect.
So this data was just presented by Dr. Isaac George at New York Valve meeting a week and a half ago at the end of June in New York, of course. And And so we did a trial, 67 patients in India with our mitral valve, and followed them for 1 year. So that was also simultaneously published in JACC. That publication came out as well last week, and so that's available. But I'll just give you kind of the overview of it. It was a single-arm prospective multi-center study that happened in India. We had 8 sites, enrolled 67 patients. Without getting into any details, I'll say that it was probably a world record on enrollment time for a mitral valve study. It happened very quickly. We had more than half of them were female. I talked about that briefly with the rheumatic heart disease, and so a good portion of them were also childbearing age.
Makes sense.
Mean age of these patients was 42, right, all the way down to 19. So a big range and young patient population, right? When we compare it to, what you see on the bottom right is a table that compares it to the Merrill daffodil valve. So that's another valve that's been on trial and then approved in India as well. We had pretty great performance. Of course, all of these patients, right, they don't come in because they're totally healthy. They are, undergoing some stage of heart failure that can lead to other end organ dysfunction or failure. So there are patients that pass away during these trials. None of the patients that had that wasn't due to the valve, though, for our study.
Gotcha.
And so, looking at the patients after a year, the graph on the left shows the mean gradient. So that's that pressure gradient over, across the valve. And that's what you see in the blue. So it drops significantly, which is a good thing, right? From their baseline, the pressure goes down across the valve. And then the effective orifice area, so that goes up, it almost doubles from where they are before they receive the valve. And so that's another good metric. It means that it's good and it's consistent over time. And so if you look at it by valve size, that's what's on the right, looking at both the mean gradient and the effective orifice area over time. You see that it's pretty consistent. Each valve size is going to have a little bit different pressure gradient, right? You're dealing with a different orifice size and different patient population, but this shows kind of what happens over time, which is all good that we see. And then, finally, 81% of these patients were in the New York Heart Association classification that's shown there in class 1. So that means that they became much better than they were at baseline. You see that? So I hope that's straightforward. So they go from class 4, class 3 down and improve quite a bit. They also do a walk test as well, and that's shown on the right. What happens with their 6-minute walk test, as well as their KCCQ score, which improves over time, quite a bit. So that's the summary of this trial, and the approval that we've received by the DCGI in India. So that's kind of the FDA equivalent there, to very shortly launch commercially in India.
Okay. Ryan, the last chart you showed, it showed that the graph with the EOA and the gradient. And then earlier you were showing a similar graph with the bar. So this is a result of the clinical, and the previous, earlier you had a similar. What's the difference between, this is just the study versus some other data?
Yeah, the bar chart that I showed a couple of slides ago was what we did on the bench over 600 million cycles. And so 600 million, if you do the math and you divide that by 60 beats per minute, so say your heart beats once every second, then that's about the equivalent of 19 years. This is only 1 year, right? And so this is 1 year, 67 patients, and what their EOA looked like.
Okay.
And again, this is an average, right? So there's a
Yep.
bunch of different sizes there. What I showed you before was just a single size.
But they both show the same pattern, right?
Yeah.
Yeah.
Yep.
Okay. And that chart looks very similar to the previous, based on the benchtop.
Yep.
Okay.
Yep, exactly.
Gotcha. Okay.
So
So,
we're still collecting data and still following this, and onward and upward, as they say.
yeah. What's the, this is really great information and a clinical 1 year study. What's kind of the next, the next step for full, 510K, or can you tell us a little bit about that?
Yeah, we have a number of things that we're working on. Like I said, so the first next step is just our commercial launch in India.
Okay.
Right, that's one of the things that we're gearing up for, right? So that's
huge.
There's a manufacturing demand for that, right?
Yeah.
And production demand, and everything that goes along with a commercial launch, right? It's very different than a clinical trial.
Yeah.
So that's happening in India. In parallel with that, we're also and have been working on preparing things for continued, I would say, an IDE pivotal study in the U.S. We did an EFS in the U.S. Before we did our trial in India, and so we're kind of taking that into a full pivotal. It's also should be on our website and not a secret. We're also working on transcatheter valves as well as surgical valves. So
Nice.
Yeah, that's our future as well. We wanted to start off and prove the polymer, right? Like I said, I mean, people have been trying to work on a polymer valve for decades.
Yeah.
We were hopeful that we would be able to get there with this LifePolymer after all of the development and time and money and resources we've put into it, and we've proven that this polymer works. And so now the next step is one that's a little bit more ambitious, and that's putting it in something that you can collapse on a stent, right?
Yeah.
And make a transcatheter valve with it.
Okay. Wow. When you accepted my invitation to do this podcast, I was pretty pumped, because this is it's kind of, I love the polymer technology blended with the therapy, and the charts and everything you went through was just fascinating. Thank you so much. It was really interesting.
Oh, of course. Thank you for the opportunity. It's a pleasure. I enjoy your podcast too. So thanks. This is a really cool project to work on.
All right. Thanks, Ryan.
Thanks, Steve. Appreciate it.